Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Frederic Bard. He's an adjunct associate professor in the Department of Biochemistry at the Yong Lin School of Medicine in Singapore. And we're going to be talking about genome-wide screening. And, I guess it's going to be dealing with the coronavirus and how it exits from endosomes and cells. And, we'll get more into that. Rich can describe it better. So, Frederic, thanks for coming. How are you doing?
Oh, thank you very much for the opportunity. And doing very well.
Okay. Well, if you would tell me about your current research and on the coronavirus and what are you looking at?
Yeah, so I think the study that you reference is something we did a little while ago about what are the host genes that are important for a coronavirus replication. One thing it's well known that viruses are the ultimate parasites, right? They are actually not even living entities, and they really need the whole machinery of the cell to replicate and to propagate. So the idea of that screen was to understand better what are the different proteins and machines inside the cell that are important for the virus. And the idea being that if we could interfere with this host machinery, we could block virus replication without having to deal with the virus protein themselves, which might be more viable.
Okay. So how would you stop the entry of the virus into cells? Because, a lot of people are talking about, the spike protein and maybe binding antibodies to it so it won't be able to allow the virus to enter. But, I'm sure it's more sophisticated than that.
Right. So definitely a lot of people are looking into antibodies that would block this interaction between the cell surface receptor, the ACE2 protein, and the viral protein, the spike. But in fact, there are many ways that one could interfere with the interaction between the virus and the cells. And so you could block this interaction, or you could block subsequent steps where the virus enters, is internalized, so goes into this membrane bound structure called endosomes. So this is the first step after the virus binds, is that it's being engulfed by the cell.
Why would the cell engulf it once it binds? Once it binds, that tells the cell that it should take in whatever material has bound to it?
Yes, that's our current understanding. The virus basically is going to cluster receptors or proteins at the surface of the cell, and that is often a signal for internalization, for engulfment. But in general, cells continuously recycle the surface protein, so they constantly engulf endocytosis, as we say, the cell surface protein. So it's very fairly common ongoing process without a viral infection, and it's possible that on top of it, the fact that the virus has multiple spike proteins leads to a sort of a clustering, and that clustering effect stimulates the cells to engulf the virus.
Why would cells engulf what's on their surfaces? Why would this be a common mechanism?
Oh, that's very important for the sort of maintenance of cells it's kind of like a maintenance mechanism, right? So the cell puts out on the surface different receptors, different protein machinery, and and it needs to recycle those regularly to in case they are being damaged, and also to regulate the way it functions. It will also constantly sample the surface and use it to modulate the surface. The cells are not always in the same state of physiology, so depending on the stimuli, they will adapt their cell surface.
Huh. Interesting. So it's recycling material. Very, very interesting. Yeah, I don't know. I didn't know that this was happening. So the virus will bind, and then the cell will engulf it mistakenly,
Exactly.
and then the virus is
Yes.
in. So I thought that the capsid of viruses stays on the outside of the cell, and just the internal RNA or DNA is injected. But is the entire virus engulfed, including the capsid?
Yes, the entire virus is engulfed, and then what exactly? So the sort of the liberation of the genetic material of the virus actually occurs inside the cells, and there's several steps have to occur before that. That genetic material is released inside the cytosol, really in the core of the cell, and can start to be replicated. So among the steps is, as we discussed, the internalization, so the endocytosis, and then there is the release from this compartment, this membrane-bound structure that is called the endosome. Then there need to be a release from that vesicle. So if you will, these endosomes are a little bit like little little stomachs that the cells sort of like, they just engulf something, but it's inside the cell, but it's not in contact with its machinery, right? With its it's a little package, and so that package needs to be broken up. And then the capsid of the virus itself need to be disassembled so that the RNA of the virus can be read and transcribed. So there are many different steps in that process.
Wait, so so normally the cell would take in material, and it's intending probably to recycle it. So it would take it in, and then it would it schedules it, I guess, for disassembly normally, because it's going to then use it for other things. And
exactly.
I mean, is there a determination of what to do with the engulfed material? Is it somehow different types of engulfed material are transported to different places in the cell based on the contents?
Exactly. So once the cell engulfs the material, it has multiple choices, if you will, for that material. So it can either send it for degradation. So it has a pathway where it sends things to some kind of degradation plant, which is called the lysosome. But another alternative process is destination is to be recycled back to the surface. So for the virus, that wouldn't be very interesting, but it's something that cells do sometimes. So it could recycle to a different place on the cell surface. And another choice could be to send it to what we call the circulatory pathway, where the proteins then enters the Golgi and so mix up with the flow of proteins that are coming to the surface. And then there is even a pathway where proteins can actually go in the nucleus of the cells from the endosome. So there are many different choices. I think what is unique with the virus is that it needs to break the membrane of the endosome to be really to move from being inside the endosome to being in the cytosol. And that aspect we think is relatively specific for the virus, and it's not well understood how that works.
Well, wait a second. So so the virus fuses to the membrane and then it's endocytosed, but what happens to the spike protein and everything? Is that ripped off the capsid, or does the entire thing intact get endocytosed? And I mean, does it pull membrane with it? I mean, do we know?
Yeah. So the idea is that the whole virus is endocytosed, and once it's in the endosomes, something that happens all the time with endosome is it acidifies. So the pH drops, and so this acidity inside the endosome probably changes the structure of the envelope of the virus, and it's thought to induce a fusion of the envelope of the virus with the membrane of the endosome. And so that's how we suspect that the virus is being then put into contact with the cytosol.
So what causes the drop in pH? Is it does that normally happen inside an endosome or?
Right. So that happens very in all endocytic processes. So and it's a machine called so they are so-called proton pump. So they are literally pumps that will dump protons inside the endosome, and therefore lowering the pH, increasing the acidity.
And what is the function of that? Why does that happen typically?
What does that happen?
To digest what's in there, or
it's a beginning. It's a beginning of digestion, and it's also a signal for. It's used for this by cells as a signal to on the proteins that its own protein that it endocytosed. Then they can change conformation, or they change or they can release some content. So, for example, there is a well-known receptor, the transferrin receptor, will bring inside transferrin, which brings iron inside the cells. And once the pH drops, it releases the transferrin and helps the cells to uptake that molecule. Huh. So it's a very common regulatory mechanism.
So the pH will drop down once it's in it's endocytosed, and that, I guess the virus accounts for this. I guess that helps it release from its capsid, genetic material. Or what?
Right. So the idea is that this drop in pH helps the membrane of the virus to fuse with the membrane of the endosome, and the capsid to be released in the cytosol. So one thing that is really important to realize is that after endocytosis, the virus is still not really inside. It's sort of inside the cell, but it's shielded from the rest of the cells in this little package, right? The endosome. And so it's really important that there is this release from the endosome. So there is a process of membrane fusion, and that is what is driven by the pH. And then there is, and then the capsid itself, once it's released in the cytosol, will be uncoated.
Huh. Okay. Are there viruses when they do leave the capsids on the outer cell membrane, or are they all endocytosed? Or, because again, it seemed like I thought the common story was that the virus fuses to the cell membrane and only the interior contents are ejected, but that doesn't seem like what this is.
Well, I guess it depends on viruses, yes. But for coronavirus, it's very clear that the whole, and for many viruses actually, the whole machinery is being brought inside. One of the things that the virus doesn't want is it doesn't want to disassemble too easily, right? Because it has to be sure that it disassemble its membrane in this machinery only when it's in the right place for it to be efficient. So there is also a sort of like a safe mechanism for the virus, so that it will only start to fuse the membrane and start to release its content, because that's it's no way back for the virus, right? Once you start to uncoat and to disassemble, that's it, right? For the virus, there's no return. So it has to be sure, in the quote unquote, to be inside the cell.
Yeah, I had heard also that some viruses, even at this point, they won't disassociate, and they'll somehow escape from the endosome and go to the nucleus, and then enter through a nuclear pore using a similar mechanism to which they use to first fuse with the outer cell membrane. Have you observed that?
Yes, not for coronavirus as far as we know, but yes, that there is many different locations where viruses can uncoat and so, for example, some virus integrate into the genomic DNA of the cell, and in that case, they need to be able to reach inside the nucleus. For coronavirus, however, the replication occurs in the cytosol, so it's really outside of the nucleus, and it requires this process of uncoating.
So there's 2 points at least to go after the viruses: prevent it from entering the membrane in the first place or fusing, and then you have a second whack at it. Once it's in the endosome,
exactly.
I guess preventing it from getting out, or like what would be the mechanisms by which you'd intervene here?
Yeah, exactly. So that's a little bit what we published a few years ago, is the idea that one could impact. So the virus really use this cellular machinery to disassemble, as we just said, like to get out of the endosome and to uncoat its capsid inside the cell. And so if you could identify what proteins of the cell the virus use and interfere with those proteins, inhibit them, then you could, in theory, reduce the replication of the virus. And that could be what is interesting about this approach is that it could be very general against a broad range, a broad family of proteins. So in our case, we identified this protein called VCP, valsolin containing protein, which is a sort of a machine that we think is involved either in a process of maturation of the endosome or uncoating of the capsid. Is not completely clear. But what is clear is that this protein is absolutely required at the early stage of viral sort of entry inside the cell, in these steps where the capsid has to be released and the genomic RNA has to be released inside the cytosol. So by interfering with VCP, and there are drugs that can target VCP, one can really reduce the capacity of the virus to replicate inside the cells, block it from replicating.
So do you think that the virus is sensing what's going on once it's inside the endosome, and is it anthropomorphized, but is it reacting to conditions inside the endosome preferentially, and either unraveling itself or not, or is it?
It's clearly. I think that there is this notion that the virus envelope and the capsid possibly are changing conformation. That's very clear for very virus. We can changing conformation in response to the drop in pH, to this acidification. So the virus really have this ability to sort of like activate some mechanism once it's inside the endosome. And so you could, for example, propose like, well, why don't we reduce the acidification of the endosome and prevent endosomes from acidifying, and that would, in theory, kind of like prevent the virus from uncoating. But the problem with targeting cellular processes such as acidification is that then you poison the cells themselves. And so it's a very delicate exercise to try to block a cellular function that has a normal physiological function, and but is being hijacked by the virus protein, right? Is being hijacked by the virus, and so you want to block these guys, but at the same time you don't want to poison too much the cell. Well,
once something's inside an endosome, do the surface characteristics of the endosome change to let the rest of the cell know what's in it? Could you even? I guess first of all, are you able to somehow, once a virus has entered and been endocytosed, are you able to, view this, stop the cell at this point? Has anyone captured the coronavirus, for instance, inside? Once it's been endocytosed before it unravels, or is there no time? Or is there no ability
actually, it's a process that takes quite some time, a few hours.
Oh, okay.
So it is possible to detect. The virus, as you know, is very small, so you need, and so it's a little bit hard to detect. Sometimes you need electron microscopy or a different technique, but actually you can detect it in endosome. And what we found in our research, for example, is that if we block this VCP protein, then we see this accumulation of viruses in the endosome. So it's as if the cell is uptaking more and more virus, but it's all blocked inside the endosome. And then you can really see very nice, you can use fluorescence microscopy, and you can see an accumulation of these virus particle in the endosomes.
So what do you notice about the endosomes themselves with the virus in them? And I'm sure you probably pick up some with no virus in them. Again, what's different about them?
Well, at that stage, at that very early stage of the infection process, the virus has not really had a big effect on the cells. So it's basically an accumulation of material inside the endosome. And it's just like what we think is happening when we inhibit VCP, for instance, also we're inhibiting a maturation of the endosome. So what we observe is that the what we call early endosome, so the endosome has just been formed, are not maturing into late endosome. And so in terms of molecular markers at the surface of the endosome, there's not this transition that we should typically see. Those are the main block that we see.
So, all right. So, what's the function of these various proteins you're talking about? You know, I kind of been asking my questions, but I mean, go ahead with what your theory is and what you how you want to intervene in the coronavirus.
Right. So, I mean, to give, for example, like to connect maybe with a bit of the current news cycle, right? Like people talk about hydroxychloroquine, right? Which basically it doesn't act on the virus itself. It's not thought to act on the virus itself. And one idea is that it interferes with endosomal maturation, or it interferes with the pH dropping. It might have multiple effects on cells, but this is one of the theory of how this compound works. And so similarly, we could expand this idea of interfering with our cells, right? Maybe making them a little bit sick for some time, just for the time for the virus to sort of to block the virus and make it being degraded inside the cells instead of allowing it to be released in the cytosol and replicate. How do we do that is not completely clear at the moment, but perhaps inhibiting that VCP protein I've been talking about could be a mechanism where the cells engulf a lot of virus, but it doesn't let the virus replicate. And so basically it mops the virus. This will just break the replication cycle of the virus. And so the question that we really, we're trying to figure out at the moment is, can we treat cells and by extension, the whole organism, a whole person with a drug that inhibits VCP and in a way that will not make the person, sick, that would be safe enough for the person, but at the same time block the virus. And this is really the question at this stage is, can we find what we call a therapeutic window, an area where the protein, the host protein, our own protein is inhibited, is reduced in capacity to the point that it really interferes with the viral cycle, but it doesn't make the cell, too sick.
What about once the coronavirus has replicated, that it's ready to burst out of the cell? Now you have, I guess, virions sitting in the cytosol, supposedly. I don't know, what does that state look like versus, a virion or two sitting inside an endosome?
Yeah. So yes, indeed. So the virus exploit the cellular machinery at every step of its life cycle, replication cycle. So it will enter what we call the secretory pathway. So that comprise a lot of membrane structure inside the cell, a lot of compartments inside the cell. And so before it can be released, the virus has to assemble into what we call the endoplasmic reticulum, and then it's being transported in the Golgi apparatus, and then it's a lot of the spike protein are heavily glycosylated, and then it is then transported to the cell surface and released. So you can see in this very complicated cycle, what the virus does is really exploiting a lot of different machineries inside the cell. So you could, so the idea here is that if you could interfere again with this machinery in a way that is not toxic, you could basically block the virus from being released from the cells, or reduced a lot the amount. And this is a game of speed. The competition between the virus and the cell is really a game of speed of replication and speed of dispersion, right? So the more time you gain, the more time the cell has to basically engages defense mechanism. So if we could slow down the packaging of the virus and its release by the cell, we could again help the organism to combat the virus.
Are the newly formed virions, are they exocytosed, or how do they get out through the membrane?
Yes, so they are exocytosed through what we call the secretory pathway. So this first compartment in the secretory pathway is the endoplasmic reticulum, and this is where the spike protein is being made. So the spike protein is made by the cellular machinery. It is built, if you will, in this endoplasmic reticulum, and then it's transported in a membrane structure where it will get together with the sort of the inside of the capsid of the virus, right? So the really internal material of the virus together with the RNA of the virus and the nucleocapsid protein. So all of that is being assembled in compartments that are derived from the ER, and then they are being transported again to the Golgi complex. And then from the Golgi, they are exocytosed to the cell surface.
Huh. When they're exocytosed, so they're these exosomes. I mean, I guess they're packaged in exosomes, or is it called something different? And when the virus reaches the cell membrane, does it just bud off, or like?
Right. So the idea, the way we understand it at the moment, it's not really exactly exosome, but it's the secretory vesicle. It's and the idea is the virus is really hijacking again a normal process by which the cell secretes protein in its environment. So, for example, some of our pancreatic cells secrete insulin. And so the insulin in a pancreatic cell is following the same pathway. It is made in the ER, then it goes to the Golgi, and then is being secreted outside by the cell by a fusion of a secretory vesicle with the plasma membrane. And so the virus is basically doing a very similar path or travel as the insulin protein is being assembled in the ER, moved into the Golgi, and then with this inside these vesicles moved to the cell surface is being released.
When it gets to the cell surface, though, does the naked virion pop out, or does it have a membrane around it from the cell?
So it has a membrane around it from the cell, and that membrane fuses with the plasma membrane, and that in that process it basically results in the release of a newly made virus in the environment.
Huh. Wow, this is crazy. So where do you think is going to be the biggest lever to stop the coronavirus? Like what you're focusing on? Once it's been endocytosed, or is there another mechanism by which you think this will?
I think what is really interesting about this kind of approaches. So obviously there's a lot of different approaches to combat the virus, right? I mean, you have the main approach that everyone talks about, which is using the immune system to with a vaccine to create antibodies that, as we said earlier, will block the interaction of the virus with the cell. But then there are other efforts on really targeting viral proteins that are involved in the replication of the virus. So the advantage of targeting these viral protein, is that they are really not needed for the cell by definition, and so you can hit them as hard as you want, and hopefully that will block the virus. The problem is that the viral genome tends to mutate, so it's a bit of a moving target, and it can be also very different between different viruses. So what we are looking at is a very different approach, and the idea is like we'll try to interfere with cellular process, and these processes, they will not mutate, right, with the virus. They are constant. They are really dependent on the host. So that's the big advantage. The big drawback is that by definition, this will have an effect on all the cells in the body, and so it could have some toxicity. And so the idea is really to control the toxicity would be to act at a low dose, but maybe a different stage in the viral replication cycle. So you would maybe reduce the efficacy at which the virus is being endocytosed, reduce the efficacy at which the virus is being released in the cytosol, reduce the efficacy at which it is being packaged and sent outside. And if you can hit at these different points inside the cell, maybe the cell can cope. I mean, the idea is that the cell will be able to cope with these different, maybe a combo of drugs. They will be able to cope with it, not exhibit toxicity, but being very efficacious to block a viral cycle. One has to realize that the virus is really gathering all the resource of the cell to make a lot of virus very quickly. And this is essential for the infectious cycle. Yeah, things have to happen in mass and very quickly. So if you can sort of block this, like really reduce the efficacy of these processes, you don't necessarily need to block completely replication. As long as you decrease it by 80%, the immune system will then be able to take over and wipe out the rest of the virus.
Well, again, I thought that the viruses would replicate inside the cell and then they burst it open, they lyse it, and then they exit that way. But it sounds like you're saying there's an orderly exit of the really important virus.
We think that for coronavirus, it's a more, or you're completely right. Some viruses are inducing a cell lysis, and coronavirus at the end of the cycle does induce lysis of the cells, but by and large, it really it doesn't. This is really a late event. So the cells produce a lot of virus before it's basically getting too sick and being killed. And this is really important for the virus cycle, right? Like the virus wants to be completely packaged in a nice new infective particles before it kills the cells. So that's maybe like one way we can understand why the virus is using this orderly process, as you say, to produce a lot of particles. And so this is because. So the first wave of production of virus is really it doesn't induce cell destruction.
Everything I thought I knew about viruses is now there's at the very least expanded behavior, if not new behavior. So do you think that so you're saying with coronavirus there's an orchestrated or orderly exit instead of an explosion of the cell, right?
Yes.
But okay, so if that happens, then what? What happens to the cell? Do all the viruses leave? Do some stay behind and finish sucking it dry? Like what happens to the cell?
Eventually so the virus will just exploit the cell's resources, right, to make more and more virus, and there is literally many thousand-fold. So for one particle that enters the cells, there will be hundreds and thousands of them that will be generated by the same cell. So it's really the cell is being transformed in a factory for virus. And so these viruses that are being released, obviously they go and then they infect other cells. And then you have this explosive cycle, if you will, where more cells are making more virus. And then for a while, you're basically brimming with virus, which is why, when people do this swab, they can detect very easily the viral genome because the cells are basically releasing a huge amount. So you start your infection with very little material, and then it being produced in huge amount by our own cells, And so the speed at which this process is occurring is really important because the virus needs to being dispersed in the environment before the immune system takes over and basically kills infected cells. That's one of the main way of getting rid of the virus.
How does the immune system recognize that a cell has been infected?
Yes. So there are different ways. So the first cells get infected, they tend to they can detect that they are infected, right? And so they release signals such as interferon gamma, and on top of it, they will sample the proteins that they have inside, and they will start presenting at the surface fragments of the viral protein. So remember that the cycle of infection takes many hours, right? So in that time, they start to produce at the surface, to expose at the surface with this MHC complex, bits and fragments of the viral protein. And now the cells of the immune system can detect this, and they detect that this is not normal, right? These things we've not seen. And on top of it, neighboring cells that have been killed or cells that are being infected are releasing this sort of stress signals, this danger signal. And so the whole immune system is sort of like being activated by these dying cells. And on top of it, it can recognize at the surface of cells that something inside this cell is not normal. Something is happening.
I guess maybe another way to impinge the virus is to cause the cells to signal very actively early on with virus fragments or other things like that,
Indeed, that could be a way to speed up the recognition. I'm not aware that anybody has come up with a way to sort of improve the viral defense mechanism. But if we could do it, that would be a great way to sort of boost up the defense. Basically, I think when we think of defense against virus, we think about immune system and vaccination. But in fact, there are many layers in how our cells, our body, are defending us against the virus, and we could, as you suggest, exploit this other mechanism. We could try to boost them up as well.
Well, what specifically you're going to focus on? I guess there's a lot of different targets, which is great. But
right.
Your work just understanding this, or are you actively looking for a mechanism?
Yeah, we're trying to. Our lab is really like focused on molecular biology, right? So we provide like targets, if you will, and now we're starting an exciting collaboration with a biotech with different people in A-Star in Singapore and with a biotech in England called Foremost, and they happen to have a drug that is active against VCP, that protein that is important for viral exit from the endosome. And so the idea is that we want to try now if we can sort of optimize the concentration of VCP, whether we can combine it with other non-drugs to have a cocktail. And that's where really we're trying to move forward. That's the direction we're taking.
Okay. Well, very good. What's the best way for people to find out more and to read papers you've written and to keep tabs on your work?
Oh, yeah. Read papers, and then if we find anything exciting, we'll definitely communicate about it. But
Okay. Well, what's the best way for people to follow up? Is there a website they should go to, or how can they find out more specifically about what you're doing?
Yes, I have my lab website is linked to A-Star, so you can find it I'm actually working at A-Star in Singapore and in the Institute of Molecular and Cell Biology. So you can definitely find my website there, and if people have any question, I've already been contacted with in regard of this work by email, so people definitely contact by this.
Okay.
So it's Fred Bard fbard at imcb astar.edu.sg.
Well, very good. Well, Fred, thanks for coming. I really appreciate it.
Thanks a lot for the opportunity, Richard.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.